In the interest of time, we'll move ahead now to module number 3. So let's begin module 3 of Cure Duchenne's webinar, Duchenne Clinical Trials and Drug Development, and I will hand it over to Dr. Barry Byrne.
Thanks very much, Sharon, and thank you all for sticking it out through the end of this webinar. I'm going to put a cap on all this by trying to address how we can maximize participation in clinical studies, and it was mentioned by Petra and by Dr. Marks, the really selfless volunteering of families to involve their children in studies are the only way that we advance the knowledge about how to determine the safety and efficacy of clinical studies. And so the pioneers that enter into such studies are it's always so much appreciated because it's an essential part of the drug development process. I'm going to have to ask you to advance the slides. These are just briefly my disclosures as an advisor and an inventor of AAV-related technology. Next, and I do have either the privilege of distinction of having been involved in this for 20 years when we first discovered that AAV vectors can effectively lead to muscle transduction. Back then, we didn't appreciate that there were many different flavors of AAV that could effectively be delivered through the bloodstream, which has a much wider effect than individually into a muscle. And this was a key discovery in the early 2000s. And then that was further enabled when we were able to figure out how to make enough of those vectors to not use just simply locally but systemically. And this is really a key enabling technology for Duchenne dystrophy, because really almost a third of the body weight is due to the musculature, and it's necessary to reach all those muscles to have any meaningful clinical benefit. And I want to go through the process of how one fully understands the new, in many cases, new information that comes to a family, often of a toddler and maybe someday in the context of newborn screening. But the average age of diagnosis of Duchenne is about 4.5. Some with milder disease like Becker may be later in young adulthood, but this is obviously a time that's very challenging to assimilate all the facts that are provided by the Duchenne community, by all the patient organizations and the umbrella organizations that consider muscular dystrophy their responsibility to serve and educate patients. So this is one example of a resource, Duchenne.com, which can provide a straightforward, meaningful background to understand how this genetically defined disease is inherited, sometimes via the mother, sometimes a spontaneous mutation in that child, and only one occurrence in the family, but not infrequently. In fact, 2/3 of the time, there may be multiple siblings, male siblings in the family that are affected. So if you go on to the next slide, just to reiterate what was said earlier to drive home that message, we rely on the DNA to formulate the recipe, as Petra referred to it, the code for making proteins. And actually, the dystrophin gene is the largest in the body, and that's one of the reasons it's subject to errors when cells are being replicated. And this impacts all muscle cells and in a way that causes cells to degenerate and regenerate and ultimately be lost. And this important structural protein shown in the lower right, dystrophin, is even though it's the largest gene, it's the second largest protein in the body, and it's subject to forces within the muscle to really be the structural underpinning of the muscle, like the shock absorbers that help the other proteins in the muscle withstand forces during contraction and that it's required for movement. This affects not only the peripheral musculature, but the heart and the diaphragm in a key important aspect of Duchenne dystrophy, particularly later in life. And it happens later in life because we have a certain amount of compensation that can occur in the skeletal muscles and even a greater amount in the cardiac muscle and diaphragm. So that lets us resist the slow degeneration of those muscles and maintain function. So if you go on to the next slide, this is where I'll just introduce the idea of the many things that can be determined through genetic testing. Classically, Duchenne dystrophy is actually a clinical diagnosis in a male with evidence by physical exam of muscle hypertrophy, sometimes developmental delay. But now with the advent of genetic testing, really one of the fastest routes to the identifying an affected child when there's a suspicion of Duchenne is through a genetic test in the peripheral blood. And only rarely is it necessary any longer to sample the muscle and look for the absence of that protein or the disruption in the muscle structure. So the questions here highlight how one identifies those mutations, how they can be used to predict the severity of the disease, and luckily in the current era, thanks to organizations like Cure Duchenne, TPMD, and the Muscular Dystrophy Association, children are no longer denied access to testing because of funding constraints. With the advent of therapies that depend on a specific mutation, there is now rationale for why testing matters, and I'll just highlight that as we go along. So the next slide. So the test helps determine the type of mutation, and without getting into the weeds of the specific individual causes, there are regions of the gene that are more susceptible to damage and errors. And so these represent large deletions which can interrupt, as Petra described, the reading frame, or that the sentence that is encoded in the DNA makes sense. If it doesn't make sense at all, it would be called a nonsense mutation. And when there's a word that's interrupted, inserted into the sentence that causes the sentence to stop. So in other words, like a period where it doesn't belong, this it doesn't make sufficient dystrophin, and that's a large proportion of the abnormalities in Duchenne. Sometimes extra material, so a piece of the DNA that's within an adjacent region is duplicated. That also causes an interruption in the gene. And then occasionally there are smaller mutations which are harder to detect, but also in effect lower the amount of dystrophin and therefore make the cell susceptible to damage. So if we go on, having Duchenne, and by now with the advent of this test and understanding the severity of the disease related to the test result, to be included in a study. So these are some of the conditions of participation in a clinical study for Duchenne. As I said, a critical part of the drug development process is the ability of patients to volunteer for clinical research studies. And so to be included, this is a very simplified representation of what sometimes is a very long, complicated list of who's in or out of a study. But to be included, you must have Duchenne. Obviously, in some cases, in some studies, it's sufficient to have the clinical features, and then there's backup information about the mutation. In some studies, they'll actually specify what type of mutation. So, for example, a drug that may work with only a certain class of mutations would require a specific mutation and not simply the clinical features of the disease. Often, the children are characterized based on the ability to complete some of the tasks that, as Dr. Marks pointed out, we want to understand how patients, how they function, how does this impact their quality of life. And so there would be a distinction between the kinds of tests that would be relevant for a patient who's ambulant versus one who's not, or one who's too young possibly to even cooperate with volitional testing. So those things have to be tailored to meet the age criteria of the patient and their abilities. One challenging part of the notion of early treatment in Duchenne is even from the time of diagnosis to the early school age years, children will gain skills, although maybe at a lower rate than their peers. And that means that a clinical study done in young children may last much longer than one done in a slightly older population where there's an anticipated rate of decline or change. And if that is the study design, you can anticipate that studies will be extended possibly for 2 or 3 years, which is a very important to consider when participating in a study. Is that too much of a burden? What impact does that have on the family and the child's participation in school and in their community? Because the studies are not often conducted at many sites around the country, but in specific locations. So it often involves travel, and that has a burden on the family. So children, again, are characterized by their functional status that may lead to inclusion or exclusion. So if the study's only for kids who have to have a certain ability to walk, then that will be a characteristic. Often now many studies require a certain concomitant medical therapy like glucocorticoid use, because adding that in the midst of a study can confound the result of the drug that's being studied. So they have to have those in a steady use prior to participating. Another point that was made is it possible to participate in 2 studies at once? That could also be very confusing. So in general, therapeutic studies restrict patients to participating in that study only at that time. If it is a drug that can be discontinued and the next study is defined what eligibility is based on how long it was discontinued, it's possible to stop one study, wait for that period for that other drug to not have any effect any longer, and then continue in another study. That's very different in the context of AAV, and it's an important thing to mention. This is a huge undertaking for any family because it is effectively lifelong. So the goal of very long-lasting therapy like AAV is that it persists, and it's likely that unless a study is designed specifically for adding a second medication to a patient who's received gene therapy, that anyone who receives at least an AAV-based therapy would be excluded from receiving another AAV-based therapy because of the immune consequences, and I'll touch on that because there are many ways that people can be exposed. So next slide. You can jump ahead. So just to go briefly through some of the ways you can access these studies. The Muscular Dystrophy Association has a care center network of about 150 sites around the country. If you go to the next slide, the mission of the MDA is to use this Innovations in Care initiative to get some uniformity among the sites, both for the 2,000 providers that staff these sites, but the many individuals who receive their care at the sites. So that's part of the MDA mission. The next slide shows you roughly where these are. They're really very nicely positioned around the country where people are, and that there are 9 regions, and every region has medical care liaisons that help facilitate care of patients and entry into clinical trials. And now in the current care center network requires the site to not only provide excellent clinical care, but to participate in research. So go to the next slide. And so I'll tell you now, as we kind of get through the last bit of this, how one finds a study. This site, maintained and mandated by some federal legislation, lists all therapeutic clinical trials, the clinicaltrials.gov. And you can see some characteristics of it on this title slide. There are 170,000 studies and also in 187 different countries. So it's not only the U.S. So if you go to the next slide, if you type into the condition or disease box, and next slide, Duchenne muscular dystrophy. And go on to the next slide. You see just this week I added this slide. You see that there are actively right now 260 studies in the entire database and 42 that are recruiting patients under the notion that they have Duchenne muscular dystrophy, and 5 studies for Becker, for example. So go on to the next slide, and I just pick one of these because it's relevant to our topic. The burden of access in Duchenne dystrophy is a study that we're conducting at our site in conjunction with the Little Hercules Foundation. This will ultimately enroll 130 individuals aimed at understanding what are the difficulties and barriers to participating in studies and access to clinical care. If you go to the next slide, this is the kind of detail you get when you drill down into the study details, and the site will list what it takes to participate, who do you contact, and this is the best portal of entry. And in fact, there's now legislation in the past few years that's required that studies post results of the studies ultimately when the studies are completed. So that's very important for healthcare providers. So next slide. Just to reiterate one of the points we raised about AAV exposure and the very promising nature of AAV therapies in the context of Duchenne, to highlight what Petra said, this is a unique DNA-containing virus that by itself requires another virus to cause disease, often adenovirus, and that's therefore the name, adeno-associated virus. The adenovirus part is a whole different virus that itself can cause respiratory or gastrointestinal problems. AAV does not by itself. But in the context of an AAV therapy, much more of the virus is given than would ever be experienced in a natural infection. So it is not merely benign as the natural virus is, but it's because of the large quantity that's given to reach all the musculature. So we have to consider that. So if you go to the next slide, this is the landscape of AAV gene therapy, and this ecosystem of biotech companies is actually helping inform one another and the field about how to conduct AAV studies and how to reach the goal, as you see the intended time to market launch. And as Petra noted, just a few months ago, the AVXS-101, the Vexus and Novartis treatment for SMA type 1 was approved. At that time, this slide was made, it was still under consideration. But you see there are many other sponsors, many of whom are working in Duchenne or related muscle diseases with an interest in developing a sustainable commercial product for gene therapy. So all the dark blue dots are neuromuscular. So if we keep going, how do we take advantage of both the opportunity of this kind of therapy and the challenges of participating? And many of those points Petra highlighted, so I'll be able to go quickly through it if you go to the next slide. So as I mentioned, one of the key exclusion criteria for participation in an AAV study is prior environmental exposure to AAV. We would not even consider those who were already in an AAV-mediated gene therapy study, but just the presence of the vector and its related serotypes in the environment can actually cause someone to become immunized, much like one receives an immunization in early childhood. The immunization causes antibodies to be formed, and this is a representation of how, in this case, the red part would be the vector and the antibodies circulating in the blood. They have two important effects. They protect us from infection, so would they protect then the muscle cells from receiving the therapeutic AAV? And we know that they in fact do. So if you go to the next slide, what happens after the first exposure is you have one kind of antibody formed here in the blue line, and then later a greater amount of the more permanent form of the antibody called IgG. If you come along, say, now to do a therapy and this primary exposure happened years ago, you would have a lesser early response, but a more exaggerated IgG response, and this can be both a safety concern and might limit efficacy. So this is why testing is done, and I'm going to touch on what the testing is for and why it's important. So we'll move to the next slide. The AAV vector can reach cells in the body that both cause a reaction immediately, so the so-called innate response, and then some late effects that the antibody-producing cells and cells that can actually kill infected cells, the T cells, are activated and come out to try to protect the body from the infection. We want to try to minimize that, sometimes through concomitant medical therapy, and that's one use of glucocorticoid, but they themselves are actually not that potent or specific in the way they're used in this context, because patients still develop antibodies to AAV and despite the use of glucocorticoid. So if you go to the next slide, we rely on these assays, and we'll skip right over this because it basically, the one type of assay assesses the ability of the AAV to get inside a cell, and then another assay, or the total or binding assay, is a different type of test in a dish without using cells as the target. And this is very reproducible and helps us differentiate between the early type IgM and IgG, and the other assay does not. So there are some efforts afoot supported by initiatives at the FDA to develop standards to make this a uniformly consistent test throughout the AAV community. So keep going about what this means for patients. If one has environmentally acquired existing immunity, that excludes you from a trial, and that, as we know so far, in Duchenne patients, we've screened almost now 400 boys with Duchenne using the ELISA total antibody assay, and found that between 50 and 60% have some evidence of prior exposure to AAV9 or a related AAV serotype. And this is probably very similar across all the serotypes of AAV, and as I said, this excludes patients from trials. If you move down through these questions, then the next slide and the next slide after that, you'll see the questions come up on the left. We want to know, is there a threshold that should be used in clinical trials, and is it appropriate? Because right now, the threshold has been set is essentially 0. And then what can we do to take someone like the subject who's from day one compared to those that might be slightly over the threshold and can use the medical therapy to reduce AAV titer, or those that have very high exposure, like someone who participated in a study like subject A, the red line, they may never easily reduce the titer with the standard medical regimen. But we've done some studies to show that it actually is possible to lower the pre-existing titer and increase the percent of patients who will be eligible. And this is a question that any patients and families have. If you can go on to the next slide. And so, yes, what level excludes patients from treatment? So if you keep going, when you think about the exposure to antibodies, it's actually better theoretically to do this early, and this is why the success of the SMA treatment particularly will be relevant in newborns who have not had an environmental exposure. But since we're mostly talking about school-age children where the exposures may have happened among peers, would receiving it earlier be better and last a whole lifetime? In some cells, maybe yes, like the heart is certainly able to retain the vector for the whole life of a cardiomyocyte, but the muscle cells turn over. So early exposure is not going to be a lifelong prospect, unless, as was mentioned earlier, there is an opportunity for editing, and I'll touch on that. So move on through these so we can kind of get to the questions. Because muscle cells divide, the copy in a non-integrating virus like AAV does not survive the division of the cell, and each copy gets less and less and less of the AAV in a muscle cell. And this happens very aggressively in Duchenne because there's cellular degeneration and regeneration that causes the repair cells to come into the tissue and replace the muscle. And the ones that come in to replace a damaged muscle have not been exposed to AAV because they live in a very quiet area of the basement membrane next to the basal lamina of the muscle cell where there's not a lot of exposure to the vector. So those are the stem cells that repair the muscle. So that is one problem. So aside from the antibody issue. So if you go on, not to give up all hope though, even though we know that and this is a study done in primates, when we do repeated muscle biopsies, you can see that the effect of growth has on the amount of gene product that's made with a systemic AAV9 in newborn primates. So this is the basis for why we began to try to study ways to prevent the immune response and be able to get multiple administrations over the lifetime. So, and that's relevant to those who have antibody in the first place. So go on to the next slide. And as I mentioned, these are diluted, so we must consider repeat dosing or figure out how to get a permanent change within a muscle stem cell. If you go to the next slide, just last week, a paper by Duchenne and his colleagues, as well as Charlie Gersbach and Catherine Wagner, published a paper about the opportunity to edit muscle stem cells using a unique marker in a mouse model in which the damaged muscle actually regenerated after the correction with this gene editing vector. So this provides some hope that this problem will be solved by getting a more efficient targeting of those stem cells. Right now, it's really only a few % of the muscle cells, but at least we know that this is possible. So this is very recent research from last week. We're getting close to the end. If you want to go on, the next slide. So what I mentioned is how can we prevent anti-AAV antibodies, and I mentioned that the first exposure is one issue, but also we want to lessen the impact of these antibodies to improve safety. And so if we go on to the next slide, with some conventional drugs, we found that we can stop the anti-capsid antibody formation. This is important both if prior exposure and to lessen the effects of the antibodies, particularly in larger patients who receive a greater dose and have even more likelihood of having pre-immunity. It enables early treatment as other approved therapies become available. Newborn screening will begin for Duchenne. And then it is an interesting opportunity to lessen side effects by doing incremental dosing to reach a desired effect rather than the notion that the drug has to be given all at once. This would be more like a conventional drug therapy, which had only in a study we recently did in Florida has this ever been shown before in humans to give repeated dosing of AAV. So next slide. We tried to study this in MDX mice, and just to say quickly, in this last column, the purple column, you can see in green, if you give AAV twice to the mouse, you get very high antibody levels, but with the presence of immune modulation in this purple, the last column, it looks exactly like the ones that didn't get any AAV at all or got AAV once. And so there's an opportunity that we think to be able to implement this in the context of Duchenne by studying MDX mice and the amount of dystrophin they made. So on to the next slide. Just to finish with the two take-home slides, we heard about how AAV can be given systemically now because of efforts to manufacture larger quantities. This also creates a new problem. We now have more immune-mediated toxicity related to the high quantities, but we can block that with a drug treatment prior to the exposure. early may be better for preventing muscle damage, but it will probably be less durable. But this would be enabled if we block the antibody response. And the last slide is that we think these same strategies can be used to prevent pre-existing to treat pre-existing antibodies, and that we have to acknowledge that there, as was said earlier, risks have to be justified by benefit. This is particularly true of all pediatric research studies. It's a regulatory requirement that there has to be evidence for direct benefit, so it becomes very important to try to reduce risk and enhance the benefit, and that's what we think the strategies are enabling. So I'll end there. We'll get to the questions. I always like to thank first patients. This young man, Will Barkowski, made this amazing figure that was auctioned at a patient care meeting. You can see he painted that with the wheels of his chair. He's the patient who, the first commercial eteplirsen patient, and then this is our team who does all the work. So I'll end there and take questions.
Thank you, Dr. Byrne. That was a really great discussion. We've received a lot of very good questions, and we're not going to have time to answer the majority of them, although you actually answered a lot of the interesting questions about pre-existing immunity and dealing with redosing. So I'll just follow up with one question that's a little bit different, and that is, what do you think is the next generation delivery to muscle tissue? Is it gene therapy? Is it something different? Can you comment on that?
I think given the body composition, the location and extent of the musculature in the body, we have to rely on the vasculature. Unfortunately, also a lot of all the organs in the body, the abdominal organs, particularly the liver and the spleen, have a lot of space in them, and a lot of the vector goes there. That's great news if you're treating hemophilia, because that's where those therapies are directed to make the secreted proteins that help for hemophilia, but it does not help in the context of gene therapy for Duchenne muscular dystrophy, because a lot of extra material, more than 100 times more than goes to the musculature goes to the liver. And that means, if you think about it, only 1% of what's given gets to where we need it. And luckily, it doesn't cause tremendous problems in the liver. In many of the current products that are being tested, they're not even expression in the liver, because, as Dr. Coffin mentioned, you can regulate the expression with different tissue beds, and so the muscle-specific promoters used in the current constructs don't express in the liver, but a lot of the vector protein goes there, and that can create a form of hepatitis, and that's been a problem in some studies. So I think we have to deal with how to work with the vasculature, because it goes everywhere we want it to go.